Hi, good day dear adjudicators. I'm Darshla Ramasamy. Matrix number 193827 would like to present my proposal on transformation effects of palmitic acid and oleic acid on polarization of murine macrophages. The main treatments used in my project are palmitic acid and oleic acid. Palmitic acid is a saturated free fatty acid which produces a pro-inflammatory cytokines and also they possess anti-cancer properties. Meanwhile, oleic acid is a monosaturated fatty acid which produces anti-inflammatory cytokines and also they possess cancer properties. According to the latest research, palmitic acid is a saturated fatty acid whose blood concentration is said to be elevated in obese patients, which causes a few inflammatory responses. They also trigger the activation of various signaling pathways that are common for LPS-mediated toll-like receptor activation. In particular, metabolic products of palmitic acid affect the activation of various PKCs, ER stress, and also they cause increase in reactive oxygen species, which is in this case ROS generation. ROS levels have cytotoxic effects, inducing activation of apoptotic pathway and inhibit the resistance to anti-cancer treatment. Indirectly, it increases the iNOS expression, which is what we really need in this experiment. Besides in research of examining the effects of oleic acid of adipose tissue, it is said that macrophages have increased arginase 1 expression. Apart from what fatty acid can create stimulus in our body, macrophages are the ones that respond to it. Macrophages polarizes in different subsets and they possess a specific functions. Different stimuli activates the generation of M1 and M2 macrophages. M1 and M2 macrophages differ phenotypically as well as in their release of pro and anti-inflammatory cytokines respectively. Well, these slides, as we explained earlier, nitric oxide and arginase is an effector of the innate immune system, which will correspond to iNOS marker and ARG1 marker respectively. The objectives of my project is to formulate and verify the conjugation of PA and OA with free fatty acid BSA using UV-Vis analysis. My second objective is to determine the morphology and phenotypic effect of PA and OA induction towards RAW 264.7. And my final objective is to determine the transformation of RAW 264.7 into M1 or M2 differentiated macrophages after 72 hours of culture treatment under flow cytometry and immunofluorescence. Well, this is my problem statement for this experiment is to evaluate on the transformation effects by individual induction of palmitic acid and oleic acid induced on murine macrophages in vitro is still scarcely being investigated. This is my hypothesis. While palmitic acid is said to be even more capable to transform RAW 264.7 into M1 macrophages than oleic acid in order to express M1 surface marker. In the future, palmitic acid has the potential to exhibit anti-cancer agent that can induce the transformation of RAW 264.7 into M1 naturally, which will reduce the use of using drugs to treat cancer, which means we use more to immunotherapy method rather than using chemotherapy method. Well, in order to perform this experiment, these are the methods. Well, firstly we have to prepare the treatment, which are PA, OA, BSA followed by UV-Vis analysis, and also we need to do MTT assay. The fact of doing MTT assay is to test the toxicity of the cell. Next is AOPI. Through AOPI, we can know the cell's viability, and then the cells are tested by treating the RAW 264.7 with PA and OA. So in order to determine their morphology, we can always look at it through microscopy. And then we can also verify the transformation through immunofluorescence and also flow cytometry. So I'll explain about the procedures to conjugate PA and OA with BSA. Firstly, about 100 millimolar of PA or OA is added with ethanol, and then 2% of free fatty acid BSA in DMEM with 10% of FBS is added, and then they are conjugated of 500 micromolar of PA and OA with free fatty acid BSA. This solution will be sonicated and then filtered. Next is the expected morphology, which satisfy the objective 2. This is upon treatment, the outcome upon the treatment, which is whereby if it's M1 phenotype, it's more flattened and porous. Meanwhile, M2 phenotype, it looks more elongated, the cells. And then this slide is about the optimization measures for the treatment on macrophages. The temperature shall be maintained at 27 degrees Celsius. There will be 6 replicates of treatment to observe, each at 24 hours, 48 hours and also 72 hours. Then before plating the cells, there should be a small calculation to be done in order to make sure the right amount of cells are plated into the well plate according to the formula. Well, this slide explains in detail for objective 3. 2 replicates of 6 well plates were used to observe the cell transformation for immunofluorescence and flow cytometry. They are incubated with antibody, which is anti-Inos FITC for Inos marker, and also CD206 for ARG1 marker. We also use DAPI in order to stain the nucleus, and then the both plates will be run for immunofluorescence and also flow cytometry. So these are the 4 expected outcomes in this project. Which is, firstly, you will see about the UV-Vis analysis. In order for UV-Vis analysis, the palmitic acid and oleic acid conjugated with BSA will exhibit 2 different peaks similar to the individual component of solvent, which is PA, OA or BSA. Initially, PA, OA and BSA will have different peaks. Then right after PA conjugates with BSA, it will form another peak which is higher than the initial peak of PA or OA, which is taken individually. And then it's about MTT assay. More than 50% of cell viability loss when the cells are being treated with 1 millimolar of PA or OA with BSA. And then the third is AOPI. When treated with PA or OA with BSA, green nuclei and intact structure will show the fluorescence. So the treatment on PA and OA towards a raw 264.7. Through this morphology observation, raw 264.7 will differentiate into M1 morphology by showing fluorescent cytoplasm when it's treated with PA. And then this differentiation, it will further be confirmed using Inos marker via immunostaining. Okay, so this is my Gantt chart for my entire final year project. And then these are the reference. Thanks a lot for the adjudicators for hearing at my proposal. So I hope I've done my best, and thank you.